presentation, and we would like to invite our respected Professor and Head Department of Seed Science and Technology to welcome the gathering.
Respected Director, Seed Center, respected Dean, School of Postgraduate Studies, respected External Examiner, Dr. K. Ratnavelu sir, and my dear scientist professors, associate professors, assistant professors, and my dear students from both online and offline. I wholeheartedly welcome for this public dissertation and guest lecture, public dissertation of Kavita, and the guest lecture that will be delivered by our external expert, Dr. K. Ratnavelu sir, Principal Sciences and Head of the CICR, Central Institute for Cotton Research. And he is based on our request, sir, without any hesitation, immediately accepted for giving the guest lecture on implementation of PVP legislation and dust testing of cotton and ICR and SAU system. Sir, wholeheartedly thanks for immediate acceptance for delivering guest lecture, sir. And this type of guest lecture we are arranging along with the final Viva Vice dissertation. Based on guidance of our Dean Postgraduate Studies and our Honorable Vice Chancellor Madam, based on their advice only, we are arranging all the lectures, all the final Viva with the along with the guest lectures, sir. So it will be useful for our students, sir. So I wholeheartedly welcome all the students, scientists, both online and offline, for this wonderful lecture. Once again, I welcome you and thank you.
Thank you, ma'am. Next, we would like to request our respected Director, Seed Center, Tamil Nadu Agricultural University, to deliver introductory address.
Good morning, one and all. It's really very happy moment to stand before you for this program. My warm welcome to Dr. K. Ratnavelu, Principal Scientist, Seed Technology, ICAR Central Institute of Cotton Research, Regional Station, Coimbatore. I also say good morning to all others. Dr. N. Sendil, Dean, School of Postgraduate Studies, Dr. V. Manonmani, Professor and Head Department of Seed Science and Technology, Dr. Jerlin, who is not here due to ill health, the professors and all other students who have come over here for this lecture as well as the Viva program. So Dr. K. Ratnavelu sir is our senior. So he is known to us, I don't know for how long. So he has been with us. Actually, during I think 2003, I had gone to IRI for a PPV FRA training program. At that time, sir had come. That was my first introduction with sir. So from almost for 20 years, he has been with us. Any important aspects, we will go and discuss with him. And he has specialized in this PPVA and FR Act. Other than that, for the student thesis, we most importantly, we will suggest him as the external expert. Sir will go through all the thesis and then he will give such wonderful comments. We can say that it will be a constructive criticism. He has never given any destructive criticism. So the criticisms has been very useful for us to upgrade ourselves. So in the morning we were discussing with him. He said that he has corrected almost, evaluated almost 34 theses from Tamil Nadu Agricultural University. We should give a ovation to them. So some people we will send the thesis. There will be some comments, that's always welcome, but still sometimes it will not go up to the requirement. It will cross the requirement. It will be sometimes we will feel very awkward to see some comments from our own colleagues. If it is valuable, it is okay, but otherwise we feel that it comes with some vendetta. So, but sir has been consistent. He will go through each and every page, make corrections in the text itself, and the remarks will also be very useful to us. At least 5 theses of mine has gone to sir. In the next campus he is. So if we need any help, we will directly go to him. If we require any details about the cotton varieties, we will immediately call him. Anytime he will be lifting the phone and then giving the required information. Recently, we conducted the think tank also. So in the ICR sector, sir was there to participate in that and then give his valuable comments. This is a very warm program for us to have him amidst us. So as per sir's specialization, he is the one of the members who the implementation of the PPV FRA 2001. So for cotton, that is the main center. Sir has been almost for 20 years in this sector. He has undergone a training, international training program on plant variety protection and dust testing for the Indian experts. That was in National Institute of Agricultural Botany, Cambridge, UK. That is one of that is a great recognition that has been bestowed upon him. He has handled almost 19 projects, published 55 research papers and 29 popular articles. He has participated in many seminar conferences and training. Besides publishing technical bulletins, book chapters, and books as well, some 5 books he has published. He is a life member of the Indian Society of Cotton Improvement, CIRCOT Mumbai, and he is a life member of Cotton Research and Development Association, CCS. HAU ISAR, and he is also life member of Indian Society of Seed Technology, IRI, New Delhi. So I think we are all very eager to listen to his lecture, because this is an important act, that is PPV and FRA Act. So once again, I thank you so much, sir, for being amongst us. So in spite of evaluating the thesis and then participating in the Viva, you have taken the additional responsibility of delivering this guest lecture. Thank you so much, sir. So I wish to thank our Dean PGS for constantly helping in the the knowledge sharing among the students with the chief guests from other places. So this is a great occasion for the students to listen from chief guests with various specializations. So with these few words, thank you so much for giving me this opportunity.
Next, we would like to invite our beloved, most respected Dean, School of Postgraduate Studies, to offer opening remarks.
Good morning to all. I'm really happy to be here in this particular guest lecture. Welcome, Dr. Manon Mani, Head of the Department, Seed Technology, the Director, Seed Center, Dr. Umarani, and today's speaker, Dr. Ratanavel, to accept our invitation and ready to deliver the lecture today, and all faculty members, students, and those online viewers. I'm really happy that Seed Technologies are always in front runner on organizing many of the lectures, mainly for inviting experts. Today is the only special thing because Dr. Ratanavel sir is one of the leading Seed technologists. Always he support TNAU activity. Any activities he immediately respond and he support, and he is one of the strength for Seed technology also, because as Madam previously mentioned, many of the thesis he evaluated, and he continuously giving the technical input and also continuously supporting the department to grow this department in the level, and he is one of the person who is always positive mindset and help the department of Seed technology. And here why we are encouraging this guest lecture is because every time we invite external examiners for the Viva for the PhD students. That time they come for Viva, and they if university is interested, our vice chancellor in particular, interested to have a lectures from the external experts, because especially for today's speaker, Dr. Ratanavel is his whole service in cotton, and is mainly specialized in the dust testing, and also the mainly working in ICR system and coordinating with many SAUs. So his rich experience in this field definitely will help the students to learn many things. And mainly I want to request the students, please after end of the lectures, please ask questions, get clarification from him. It is a rare opportunity for you to discuss with him in this hall, and you can make a deliberation and have a discussion, because Nowadays, you know that plant varietal protection is gaining momentum, and separate PPVFRA authority is there, and they are every varieties, every hybrids, when they either from public sector or private sector, they are registering. So when they registered, then the major thing is DUS is the key character, because unless DUS characterized, you cannot register in that. So in that, he worked more than 3 decades in the cotton, especially on the cotton, how to handle the varietal registration when you go to PPFRV. Even before establishment of PPFRV, he is working on dust testing. Am I correct, sir? So before establishment of PPFRV, he worked in the dust testing system. So the authority is established later. Then they try to implement whatever the things, dust testing protocols which are there in the ICR system. They adopted and tried to formalize the things. So that much of great knowledge he is having. Another thing is going to retire shortly this year. So it's a very huge loss for not only ICR system and also seed technology department and TNAU, because he's one of the very known person and very cordial person which always helping us. So it's really, sir, we are missing your service for mainly in Tamil Nadu Agriculture University. So this online, these lectures been recorded, and you can also been watch the later, those who are missing the lecture, and also the students who are attending the lecture. I request you to have a more interaction. That will be helpful for your knowledge sharpening, and also the speaker also will be so happy if you are interacting with him, because he is so much knowledge on 3 decades on dust and cotton. So if you interact anything, he is so happily he will reply your queries. Okay. So with this, I really thank Department of Seed Technology for organizing, the head of the department for organizing lecture, and also I thank the Director Seed Center, always keen in supporting many of the activities of the Dean Postgraduate Studies, and especially I thank our Vice Chancellor for providing the platform for many of the external examiners to deliver the lectures, and this is one of the lectures which always been remembered and always been archived in the online. People watch anytime. So this will be really a wonderful moment, sir. And really thank on behalf of Tamil Nadu Agriculture University, on behalf of the Vice Chancellor, I thank for accept our invitation and come over here and deliver lecture. Thank you for giving me the opportunity. Thank you, one and all.
Thank you, sir, for your motivating opening remarks. Now we would like to request our beloved and most respected guest speaker, Dr. K. Ratinavel, Principal Scientist, Seed Technology, Central Institute for Cotton Research, Regional Research Station, Coimbatore, to deliver guest lecture on implementation of PVP legislation and dust testing of cotton under ICAR and SAU system.
Of cotton crop in my career itself is an important and interesting aspect. When I was the student of Dr. Jacqueline Selvaraj, where there were 4 students at that time, MSc allotted, and myself, Dr. Selvaraj, Dr. Mohandan, and one Kesavan, departmental candidate. So we were there, and Dr. Jacqueline Selvaraj was able to give different crops for conducting MSc AG dissertation. So that time, my basic degree was in horticulture. It was to do in cotton. Madam forced me to do in papaya. Then I accepted it. Dr. Kesavan was given cotton crop. So that time itself, I decided that somehow I should go into the cotton crop and do some research on that. My ambition was from the beginning, I was to be in research. So I joined in the department and worked for 3-4 years as a horticulture officer. Then I resigned that job and joined in the ICR system when I was selected for ARS in 1989. I joined in 1989 in the ARS system. I was given 3 opportunities to select my crop to work in the ICR system. One is sorghum and another is cotton. The third one is sorghum. Dr. Tonapi cleverly chose that sorghum crop. Dr. Rajendra Prasad. These 2 were, though they are my batch set, they were senior to me in PhD. So they selected that respective crop. I was left out with the cotton. I was so happy that these 2 fellows, they never compete for selecting the cotton crop. Then I was posted in Nagpur, main institute. Then I served there for 2 years. There, my first aspect of research is those days, the number of sprays for cotton enormous, 15, 16, 17, up to 20 sprays. People, scientists also advised the farmers, and farmers also, interestingly, to save the crop, they started spraying. So this has caused a lot of havoc in the environment as well as the seed. So my first experiment on cotton was to study the impact of heavy insecticidal sprays on the quality of seed. This was my first experiment. I studied it and I concluded that I recommended that several insecticides should not be sprayed because it causes a lot of damage to the viability and storability of the cotton seeds. So that was taken care that time, some 1918 and 90. Then later on, I moved to CSR here, the regional station Coimbatore, and continuously working on various aspects, including the fiber properties. See, in genetic purity of the hybrid cotton series, 90% and 1.5% off type allowed and 8.5% self seed allowed. No? So those 8.5% of the self seed, if it is mixed in the cotton hybrid cotton fiber, so what will happen? Those plants, the 8.5% of the seeds will be germinate in the field if you allow them. So that the fiber properties of the hybrid and as well as the parental seeds will be different. So what will be the impact on the fiber quality? That was my first study here at Coimbatore. So like that, several aspects. Then technology mission on cotton, Raja was there. We have done a lot of studies on hardening, priming, and so many things. Then in 2003, the concept of plant variety protection came. That time it was not called as the plant variety protection. Simply they say dust testing, distinctiveness, uniformity, and stability. And this was the thing. Actually, honestly speaking, many were not clear about what to be done and how to proceed. This aspect took 4 years to come to a conclusion that we have to proceed like this. So we were in dilemma. 2 kilo of seed has to supply. So we circulated to all and every center for each variety we received 2 kg of seed. We were not knowing what to do with the next aspect of this program. So we realized that we have made a mistake. Then we stopped it. Then ICAR formed a committee to evaluate what is the protocol first. This task was given to NSP. Dr. R.K. Chowdhury was the program leader that time. So many task force was constituted and several discussions were there. For cotton itself, first 3 reviews we have finalized. Then PPV authority was established. It was established functional in 2008. So Dr. Nagarajan, the then ADG Plant Protection, was nominated as a chairperson of that committee. Then Nagarajan took in different perspective and he wanted to constitute a different task force and several meetings were there. Each and every meeting we were there. We were repeatedly presenting the protocol. But Nagarajan was not satisfied. Still, he wants to have some improvement. So the aim was our legislation, our protocol should match the international standard. He made that Delhi office to the international standard. Up to 5 years, it was maintained. So after that, several changes were there in the department authority, and now it is present status. So this was the history of the evaluation of this program. So 2003 to 2008, we were characterizing all the available varieties. Cotton, we have varieties, we have parental lines, we have hybrids, interspecific hybrid, intraspecific hybrid, inter-arboreum hybrid, desi cotton, and 4 species of cotton. So these things are there. But as per the PVP law, all hybrids and varieties are varieties as per this law. So we have considered the parental lines also a variety, hybrid is also a variety also a variety. Open OPV. So this has caused a lot of confusion among again the breeders, again the testing officers. Most of the universities and most of the ICAR institute, seed technology section was given this responsibility because this whole responsibility came to NSP seed. So that's why all these seed technologies were made as a nodal officer. Third party testing. Breeder should not test his own material. So that's why that concept came, that third party testing evaluation. So this law was implemented in 2008. 6 crops were selected for the first testing procedure. Cotton, sugarcane, bajra, sorghum, and 2 more crops. So 6 crops were notified for conducting the dust testing actually. So 2008 cotton was notified. Cotton was notified, diploid cotton as well as tetraploid cotton. Gossypium arboreum and herbaceum were diploid cotton native to India. Gossypium barbans and Gossypium hirsutum. American cotton and Egyptian cotton. So tetraploid cotton, these two categories were there. 2 protocols were created, published, notified. So 2008, then how many varieties we have to take? This was our confusion. So as a trial run, some 6 or 8 varieties we have taken. So how many reference varieties we have to choose for that? So again, that was the confusion. For each character, we have to choose one character. So myself and Raja, we did actually. We selected for a variety not less than 7-8 reference varieties. That was not required. That time we were not knowing. So we selected the number of reference varieties were more than the number of candidate varieties. So our trial was like that. Then how to record data? That was the problem. We have selected data sheets, data books we have selected. That book, it is a very big book. We cannot handle it. So we were not knowing how to do it. Then I devised a method, single sheet data sheet. I suggested and authority accepted it. So every time you have to take only one sheet and go to the field and take observation and record it. So that sheet you bring and file it. Like that, every character repeated. Cotton, 37 characters were there for tetraploid cotton, 32 characters for diploid cotton. So every time, 32 times you have to go. If some varieties are late or some varieties are early, again repeated visit you have to go. So for measuring the plant height, some are dwarf, some are tall. So you have to make different visits for recording the plant height. Similarly, for typical character is 50% flowering. Some are early flowering. For those varieties, we have to make a special visit to record. Some are late flowering. For those varieties, we have to make a special visit to record. So in that way, we have finalized it. Now we have customized everything. Now we are pakka. Even my RA, Dr. One Kavita was there. Without my presence over phone, I can I can tell her, and she was specialized. She was with me for past 15 years. Now she is not there, but she gained knowledge. 15 years she was with me, very sincere and dedicated. This Kavita, after this Kavita, that Kavita came. Our professor Kavita was selected for assistant professor, and that Kavita I selected. And she was Kavita and Raja, they were very sincere. And Malakodi, sometimes she was also there with me. And all our people so dedicated, they contributed so much than me for the build-up of these protocols. So this was the earlier activities happened. So now we will proceed. What is IP? Then what are all the opportunities? What are all the opportunities we have? What are all the professions we have? So what are all the difficulties we face while registering, while doing the things? This act, students must have studied, and most of the process they have studied, but slightly we will see in the different perspective. So this act, then what is the achievement in cotton at the end we will see. So this was the thing happened over the period of 2008 onwards, and Dr. Another Raja, Adudurai Raja also was with me for for 3-4 years. He was with me. So 4-5 are there actually closely associated with me. So their contribution I cannot forget. I should acknowledge their contribution in this place. This is the important meeting that we should recognize their services also. So what is actually the intelligence? We were not thinking about 2008. Before 2008, we were not. Many of us, we didn't know what is intellectual property, what is patent. We didn't know. So what to be done with our technologies? We never knew. So simply publishing one article would serve the purpose. So that was our earlier thinking. So keep on, we are evaluating, keep on our knowledge gains, and we are customized to many things, especially in the home life, social life, as well as in the academic life. So our knowledge is gaining, and we are thinking further. So what to do with our knowledge? The definition is the property created by human intellect which can be incorporated in tangible object and reproducible in the different locations. See, find out one thing. I tell you one important example. You might have heard about this Philip opener, this PepsiCo and other things, bottles, Philip opener. That technology is the more remunerative technology ever invented here, present situation. So the royalty it gains a lot. So reproduced every year, every place it is reproduced. It is different companies they are using by paying the royalty. One of the finest technology. Depending on the nature of tangibility of the IP, different type of rights called intellectual property (IPRs) are granted by the state. State in the sense country. So there is no domain for the granting IP. You can divide northern part one part or one state and southern part one state. You can divide the country in as per our convenient. As a nation also you can use, as a domain also we can separate it, and different type of IPs can be given. So another thing is intellectual property rights are legal rights which are as a result from the intellectual activity which industrial, scientific, literature, artistic fields. So once you create certain things are automatic. For example, music, automatically novel. Once it is the ISBN number, if you get copyright automatically comes to you. But whereas in plant variety, it is not so. So we have to take some steps to get it done. So the different provisions are patent, Indian Patent Act, Copyright Act, trademark, industrial designs, layout designs, protection undisclosed information, protection of database, geographical indications, plant-based rights, and farmers' right. So these are the opportunities. It is not that we should go only as a scientist, as a breeder, as a plant scientist, you can use this last opportunity. But as a scientist, as an inventor, any of the for agriculture graduates, for agriculture researchers, all these are holds good. See, copyright, you can write a novel, very interesting book, and you can get it done. Designs also we can prepare and register. Layout designs also we can, for a particular experiment we can make a pakka layout, and that layout can be permanently used. That can be done. Undisclosed information, any information generated by us, some big database, we can protect it. Geographical indication given normally not to the individual, it is given to the community and for a particular location. For example, Gundumalli, Madurai, Tirupati Laddu, then saris, so many Sungari saris and so many other things are there. Several examples we can cite. And currently we are trying to get a geographical indication for the Suvin cotton variety. Suvin cotton panji, kapas. So why we need a IPR? Incentive to the individuals and institutions, monetary return to the investment of research, breeder rights for sustaining the breeding activities. Only the commercial activity of the research. So we have invented, we have discovered certain things. So how to make it commercial? These three lines in a smaller word, we can say that it is the commercial activity of the inventor. Then for protection of Indian agriculture, what are all the provisions? Patent law does not provide method of agriculture, horticulture, any process of medicinal, surgical, creative, any living form of including microorganisms. These amendments came, and then we can do it in patent. Products obtained from the chemical process, innovations from the biological research. So these provisions are not there in the patent law, but the procedure for applying patent, the procedure for applying plant variety, both are almost same. So how these law, the genesis of the law came. Actually, WTO 1995, 1994 GATT, in 1994, GATT was there. In 1994, our economics department, when this GATT was announced, our economics department, one Dr. Balasubramani, whether Bala Subramani Rudrasamy, he is there in service or not, I do not know. Economics professor. He was that time assistant professor. He purchased this GATT agreement book with 2,000 rupees those days. So we were looking those papers. We were not understanding anything. What is there? Because all these legal terms and economic terms were there in that book. So we were not knowing that the draft I have seen. He purchased it personal with the personal interest. So nothing was understandable. So then they changed the name to World Trade Organization in 1995 with 131 countries. India was very reluctant to sign in that WTO. Uruguay round only that discussion India signed. Dr. Subramaniam Swamy was made as the nodal officer for the implementation of GATT those days. Dr. P.V. Narasimha Rao was the Prime Minister that time. So it came like that. So in that GATT agreement, Article 27.3b gave a provision for the creation of one new law as per your desire. That gives a provision to have your own law or adopt the international method. So our country, because a lot of variability is there, a lot of opportunities are, so we made our own act. So these are the patent or sui generis system or combinational system. In China, in Indonesia, Philippines, Bangladesh, dual system is there. Either you can go for patent, either USA also it is there, patent system in some of the German, Switzerland and Norway, I think. Here, some European countries, we have provisions for patent as well as PVP legislation. You can, or combination of both. This system is exist in many countries. But we have followed patent is a separate system and plant variety protection as a separate system. So why we have selected for the sui generis system? Need to incorporate equity concerns, right of farmers, right of village community, right of researchers. So we have to keep all these things in mind and greater flexibility in regard to protected genera, species of varieties, level and period of protection, sustainable development of agrobiodiversity, benefit sharing arrangement. So these arrangements we have to take, and we have a lot of variable materials. So all these were kept in mind and the sui generis system was developed. Our system only, farmers need not pay for the use of the protected seeds. But in European countries, farmers has to pay royalty to the breeder for using the protected varieties. So European, especially German people, they want to have a collaboration with us. Their main focus is that we will invest in India, we will bring our varieties here, provided you should remove that farmers' rights. That farmers are not allowed freely to use the protected varieties. So this was the condition, but our government never allowed. But some of the dialogues are going on still. I think 4 rounds we have discussed on these issues, and on all the 4 rounds, the end result was only their demand was to remove the farmers' rights to use the farmers' protected variety. But our demand is that we will not remove that to farmers. But if we want to bring, their concern is that if once OPV came to India, then farmers are naturally they will be using now. So their concern is like that. So India is not ready to remove that farmers' rights. Then the objective of law, they enact to provide the establishment of the effective system of protection of plant varieties. And another objective is to recognize the protect the rights of the farmers in respect of their contribution made at any time of conserving, improving, and making the available genetic resources. Farmers have conserved their materials. Several farmers, even today, best example one can say you can say. Then Virupachi, Pattivirambatti, Rajaram and their family, and many are there. And Ammalvar also one of the conservator of nature. So these two were given. Nilajayaraman was recognized. Then Pattivirambatti people also, that family also recognized by the PVP and given 10 lakh rupees from the gene fund as a token of appreciation and as to further movement, and a special project was given Virupachi banana for that. So like that, we have to encourage the conservator as well as the farmers coming for the protection of variety. Accelerated agriculture growth on one aspect, facilitate the growth of seed industry in the country, which will ensure the availability of high quality seeds and planting materials to the farmers. So how this act will encourage the seed industry? A day will come that all the varieties available in India, first crop would be the cotton, that all the varieties available in cotton would be with protection. So you won't get any seed in the market without a variety, without a plant variety protection. Those days only you will realize that what is the importance of plant variety protection. Then breeders' rights. You would have studied in class, so many rights are there. Breeder can do research, can invest. Breeders shall enjoy the professional protection. Actually, how applications are made. One variety is ready, then variety should attain the level of variety. Before it becomes variety, what it is a line, breeding line. Before that, genetic stock. Before that, jam blossom. So keep on, you are improving. When it will come to a level of variety, when it is tested in multi-location with uniformity, all these except distinctness with the uniformity and stability, that variety is called as a variety. All India Coordinated Research Improvement Program. That is called a variety PCU test usage. So once that level of variety comes, then it is eligible for registration. So the breeder can register even before the level of becoming the variety and enjoy the professional protection. Breeders' right will help to stimulate further research, develop new varieties, encourage growth seed industry, ensure quality seed to farmers, accelerate growth in agriculture. So these are all some of the point. Then researchers' right. So researcher can conduct research, conserve the seed, save the seed, share the seed with other researcher. These are the facilities available. Then farmers' right. Any protected variety he can cultivate, he can multiply, he can store, barter, sell in the market. Again, farmers' rights continues. Then communities' right. Recognition of the role of traditional communities. Nel Jayaraman, for example, for contribution of village local communities, then compensation to be determined by the PVP authority for deposited in the gene fund. See, if the procedure is, I am selling you to one protected variety, that variety fails in the field. So you are, as a farmer, you are asking me the compensation. Where you will ask, to whom you will ask? Question. You have to go to the court, local court first. You have to file a case. That case, I have to justify my contention. Then that case will go to again PVP, plant variety protection. Then they will give direction. If really the crop fails, then I have to be compensated. You farmers have to be compensated by me. So this was the condition. If anything goes wrong in that process, then I should approach a court not lesser than the high court. Then I should go to high court and contend. So this was the procedure. So whatever that compensation given by me to the farmer, one portion will go to the gene fund. So in that way, for gene fund, several ways of income generation is there. So this is one of the way gene fund is generated. Then what is jam blossom? all races constituting the primary genetic variability, including the secondary tertiary gene pool. Whether we can register, protect germplasm, not possible. Germplasm is not a variety. So as a scientist, as a researcher, where you can use this germplasm, you can register the germplasm as a genetic stock with the NBBGR. So what is that method? There is a procedure for registering the germplasm. Go for multi-location testing, establish your character, then germplasm registration committee will be there in every institute. DNA also must be there. Breeding department, there must be a germplasm registration committee, and committee will visit, convince about your claim, and they will recommend to be registered under as a genetic stock under NBBGR. So this was the procedure. For microbial thing, there is a institute at MAU. Genetic stock you can, as a microbes you can register there. Then land races are the races used by the farmers. It's an improvement over the germplasm through selection process. So people have worked on that. You cannot identify who has worked on that. Several farmers over a period, they will work, keep on selecting. Selection pressure only that land races developed. These are those varieties of crop which have undergone minimal selection from farmers. So farmers keep on selecting. One period you can realize that I have seen my young age paddy field farmers, they used to collect only the good earhead for sowing in the next season. So that is the real selection actually by the farmers. So everywhere, even bajra, good earhead, they will select and they will select the seed and go for next season sowing for sorghum. So like that, farmer selection were there. That they do not know that they are doing some breeding activities, but still they did it unknowingly or knowingly. So Biological Diversity Act says a primitive cultivar, landrace is a primitive cultivar that was grown by ancient farmers and their successors. So farmers selected one variety and given to other farmer, like that over generations they have used it. Then folk variety came, a traditional variety which is known to the farmer, but essentially not as a as much agronomically evaluated as the farmer's variety. Agronomically, these things were not known to farmers. Simply, they know that there is a variety, they will grow it, and whatever the yield comes, they will enjoy it. They don't see that this variety should give 5 quintal yield, 10 quintal yield, like that. So whatever that variety yields, that they enjoyed it. Folk variety, then Biological Diversity Act. According to Biological Diversity Act, is a cultivated variety of plant that was developed, grown, exchanged informally among the farmers. Again, it was an informal way. People barter, go and keep on spreading their variety through mouth to mouth extension those days. Then they will give that the seed, then they will get benefit out of that. These are the, see, basically, I tell you one thing. For all these plant variety breeding and agriculture resources, the topmost act is the Biological Diversity Act. We all comes under the Biological Diversity Act. Since that act was not fully implemented, we are having some exemptions and we are having some deviations over that, including the whole of ICAR, including the all SAUs, including all the agrobiodiversity zones, hotspots. Everything comes under the Biological Diversity Act. That act, to my knowledge, it is not completely excised. So that's why we have some exemptions here and there. Even to move microbes from one block to other block, we have to inform to the Biological Diversity Act. For using the land races, for using the traditional varieties, seeds for the breeding purpose, for other purpose, other improvement over for cultivation purpose, we have to inform to Biological Diversity Act. Whether they give permission or not, immaterial, but you should inform. You should have a proof that you have informed the biological authority, biodiversity authority. So this was the procedure. Even today it is followed. When the question of dispute comes over the ownership of the variety, this was the question asked, because you have taken germplasm or some other material from Dharwad or Bangalore or some other place. For example, sunflower, you can get germplasm lines from Bangalore. and you can do a lot of hybrids and development of varieties. So once that variety comes for a release notification, then dispute will arise for the ownership of that variety. Several we are seeing that in the varietal identification committee, we are witnessing several disputes like that. So in that situation, these questions will come. Then traditional variety, a plant variety identified or evolved by farmer or farming communities and being used for cultivation over the years, possibly with or without a continuous selection. Whether it can be selection may be there or not, but still they continuously they grow with it. Farmers variety, a plant variety which is either developed by a farmer or community of farmers or conserved by them for a long period. For example, one variety, which variety we can say? Bikaner Nirma, cotton variety, Bikaner Nirma, H777. This Bikaner Nirma was the farmers variety, selected and improved by the HIO, not HIO, Rajasthan, and named as Bikaner Nirma. The same farmers variety were improved by the CCS HIO and named as H777. So same farmers variety, but a different objective. People worked on different area, and they selected came with the two varieties. Has been traditionally cultivated and evolved by the farmers in their fields. Is a wild relative and land race or a variety about which farmers possess the common knowledge. So farmers knows about the variety. So this was the farmers variety. Then what is a cultivar? Then our it comes into our domain. Cultivar is a cultivated plant that has been selected and given a unique name because it has a desirable characteristic, decorative or useful that distinguishes it from the otherwise similar plants of the same species. One typical example, when you write article, in your article, knowingly or unknowingly, MCU 5 cotton variety cultivar CV dot MCU 5. Definitely you will write like that only. Cotton variety or any variety cultivar CV dot in your thesis, previous thesis also you can see right now also you will write like that only. So that means what? It has reached the level of a variety. That material line, research line has reached the level of a variety. Once it reaches the level of variety, it becomes the cultivar. It comes to the cultivation domain. So it is called as all released varieties are cultivars. When propagated, it retains those characteristics. Any plant variety, either traditional or improved under the cultivation or in this system is a cultivar. Then Biological Diversity Act, cultivar is defined as a variety of plant that has originated and persisted under cultivation or was specifically bred for the purpose of cultivation. So very specific purpose. Definitions are different, but the matter is same. Then essentially derived varieties. And this is another area we can do a lot of research. Seed group also we can take research. There no research, in fact, in this area. Breeding side also no research. Not that much research is there. A lot of opportunities are there to conduct a lot of experiments under essentially derivative. What it is actually essential? It should the essentially derived variety also should be a distinguishable variety, but those with all from the parental lines. The simple terms, clearly distinguishable from the such initial variety, conforms except for the differences which result from the act of derivation to such initial variety in the expression of essential characteristics that result from the genotype or combination of genotype of such variety. So here again essential characteristics, one term is there. What is essential? What is non-essential? Again, what is the characteristics first? Morphologically, you should define a plant first. Any plant, sugarcane, bajra, sorghum, cotton, sesame, sunflower. Totally A to Z is seed to seed. You should define the morphologically. You should define the plant. So while defining those plants, some of the characters will not change with a successive cultivation. some of the characters are subjective to the environment. Those characters which are not changed over a period of cultivation is called essential and mostly controlled by the genetics. And those characters which are subject to environmental influence are called optional characters. So here what we talk is essential characteristics. These characteristics should not be changed for any crop plants. So again, what are all the eligible plant varieties which may be registered? Not defined under section 5, farmers variety, variety about which there is a common knowledge. There is any other variety in the public domain. And without knowledge also, there may be some varieties. So people, farmers only, they know that this is a variety, but tapioca, you can popularly say rose, one rose variety is there. What is that rose? What is the name of the rose variety? No one knows. You can ask TNA also. I don't think whether there is a. It was brought from Thailand actually by some traders introduced in South Arcot, Salem, those. கேட்டிருக்கீங்களா ரோஸ் வெரைட்டின்னு கேள்விப்பட்டிருக்கீங்களா அந்த அந்த ஒன் எக்ஸாம்பிள். Then another one is Burma white. Burma white, there is another variety, tapioca. And for that also, that pedigree is not known, to me not known, but may not be there in records somewhere. Maybe that also from the, so but known to farmers. Farmers they know. So those kind of varieties you can try to protect it. Registrable varieties should have these things, all you know. Novelty, distinctiveness, uniformity, what is stability, what is novelty, that condition is there, what is distinctiveness, condition is there, uniformity and stability. Distinctiveness for essential characters, one notes, one variability. For non-essential or optional characters, two nodes variability should be there to define as a distinctive variety, and uniformity should be 100% uniform plus six off types are allowed. Okay, for cotton. For other crops may be different, and it should be a stable one. That the stability over a period of raising the crop, we can find it out. Definition for novelty, definition for distinctiveness, uniformity, all these you have studied. Again, these essential characters, it is applicable here also. Application for registration under section 14. So I will tell you the procedure in simple, and this is the lengthy one. And then application, that format form A is there, then questionnaire is there, form PV1, PV2, photograph of data of the crop, photograph of the crop, then fees. 5-6 items are required basically. What form PV1? Your university should allow you to register first. PV1. What is PV2? Your breeder should allow you to register. They are giving permission to you because you are an applicant. You are making an application in the name of TNAU. So I have done application for 6 cotton varieties of TNA also. Totally, I have done 96 varieties applications I prepared, I submitted, and presently 6 varieties I got professional registration for cotton varieties of universities, our Nagpur center, our Sirsa center, our Coimbatore center. All these things I only made it as a nodal officer. So what I should take in the name of ICR, I should make an application. All rights will go to ICR. Then ICR should permit me. ICR should permit the signatory also to sign in that application form. This is form 1. Then form 2. Breeder may be sitting somewhere. So breeder may not be knowing what kind of application we should prepare and things. As a nodal officer, I am doing on behalf of ICR, on behalf of institute. So I should get the consent of the breeder. Breeder will give the rights to me to prepare the application and the head to sign as an authorized signatory. So he will sign on behalf of them in that application form. So this is one thing. Then genealogy of the variety, pedigree of the variety, all these details will come in the technical questionnaire. Then required fees we have to submit. Then photograph, distinct all the morphological character it should express. All these things you should submit. Once you submit, if that application is accepted, from that date, that is the provisional registration. From that date onwards, that variety is provisionally registered. That acknowledgement will be communicated to us. So we should keep. Then for getting the real certificate, it will take another 3 years. The Plant Variety Protection Authority, that provision given to them is minimum 3 years to issue the final certificate. Sometimes it may exceed because test may failure one time, one season, some centers may not conduct. So this kind of logistical as well as seed may not reach, technical lacuna may be there. So minimum period is 3 years. So denomination, what it is a name only. So what kind of, it should not affect the feelings of any society, a community. Meaningfully, it should convey. Then National Gene Fund, I said, constituted from benefit sharing proceeds. So benefit sharing is another area. Royalty, once variety is protected, how come royalty comes to PVP authority? Benefit sharing, how come it's when there is a dispute, then arbitration we can go to PVP. PVP will give you a judgment that you should pay this much. That sharing will be there. Maybe that parental business I told you now. So that issue will come here. Then royalty. Once the variety is registered, that breeder who is multiplying and selling the seed is the registered breeder with PVP. So every year while renewing the certificate, you should pay the royalty 0.5% to the and the sale proceeds. Whatever the sales are there in that seed, 5%, 0.5% you should remit the amount to PVP. That will come to the gene fund. Then communities compensation. So while giving a company will give one village because of the crop failure. So that also there is an amount. Then contributions from different breeders. And this will be utilized for breeder farmers communities. And that fund will be utilized as per the wish of the Plant Variety Authority. A new variety shall not be registered if not capable of identifying such variety. So these are the some criteria that for rejection of the application. These are the criteria likely to hurt the religious sentiments, different from every denomination. So same name is given, feeling of the communities, the feeling of the religion. So those things will be there, and accordingly that variety will be provisionally registered. That 2008 it was established. There were that structure you would have studied: chairperson, registrar general, registrars, deputy joint registrar, deputy registrar, then technical officers, then financial section controller, financial advisor, accountant, and financial people. That wing will be there. So different DUS test centers. As of now, after the establishment of authority, 142 crops were notified for registration purpose. And the registrars are powerful and they can make decisions. 3 registrars are there, one for agriculture, one for horticulture, and one for farmers' rights. A different procedure, how to again mandate of authority, registration of plant varieties, characterization, documentation of varieties. There is a register. I have seen there was register, very big register. So they will make proper entries. Professional providing compulsory cataloging facilities for all plant varieties, ensuring that seeds of all varieties are registered, made available to farmers. So here the compulsory licensing comes. Collection and comprehensive statistics on plant varieties, maintenance of national register for plant variety. So at NBPGR, gene bank is there. So whatever the varieties we have collected, cotton we have originally we have collected for 240 varieties and we have deposited the seed. Right now we have 180 varieties. Varieties apart from that we have parental lines also. And in BT varieties, BT hybrids, we have nearly 1,500 BT hybrids. Those hybrids are in the seed chain. Those companies are interested for having their registration. They came forward, they made an application and it was tested and certificates were given. Only those BT hybrids. GAC, permitted by GAC for mass cultivation. They are not notified varieties. BT cotton hybrids are not notified. Then in 2001 onwards, BT hybrids and BT varieties are under notification. Then the plant variety registry, they do NPPGR, I said, number of registrars required, the regular national register of plant varieties kept at the headquarters. We have seen that variety. Then application, that procedure I told you, no? Person who can make application. So eligibility of the individual, scientist, farmer, group of farmers, community, they can make registration. Acceptance of application or amendment. So they have right. They are actually plant variety authority is a statutory body, not answerable only to the parliament, not even to the agriculture minister. They will submit their achievement to the parliament only. Annual report they will submit to parliament, not for the agriculture minister or secretary to agriculture. So straight away answerable to parliament. So they have a lot of powers. They can make amendment, any type of amendment. Several amendments were made. Advertisement for advertisement of application. So once application is received, published in the plant variety journal, any dispute is there, 3 months period is given, you can make a contention, you can argue, you can file a case. So many provisions are there. Total period of validity shall not exceed 18 years, 15 years for vines, permanent trees and annual crops. Why 15? Actually, you should have one question. Why 15 years is kept? Why 15 years? Tell me someone, if anyone knows. Why 15 years? Why not 20 years? Why not 10 years? For notification validity also 15 years. State government, if your variety is notified before 15 years, they won't give subsidy. Why? Why that kind of criteria is followed? There is a technical reason that after 15 years, that genetic makeup will not stand. After 15 years, there is an ample opportunity of making lot of types, a lot of rogues will come. So that purity will not be there, genetic uniformity will not be there, diverse stability will not be there. So that's why they kept it as a 15 years as a life cycle of a variety. So from the notification point, they have taken these 15 years for registration also. Many countries 15 years, some countries 20 years. Then compulsory licensing, when you have a demand for a notified variety or registered varieties, then if you don't produce a sufficient quantity of seed, then it will be automatically license will be given to the third party. UPAV International Union for Plant Variety and New Varieties for Plant. So we have a UPAV, European, Geneva is the headquarter for UPAV. UPAV and WTO, the director general is same. UPAV, here I think CEO, WTO CEO, their director general. So same person will operate these two offices. So these are all about UPAV. Then our cotton guideline came. So the varietal profile is Hirsutum. This is actually a little old data. We have some more new additions. So that previous PowerPoint I am doing. Hirsutum varieties 177, Barbrans 5. Barbrans we have to add 3 more. We have released 3 more varieties. Then Hirsutum, our office, we have released 8 varieties, new Hirsutum varieties. Herbaceum we have not released. Hybrid H into H, intra-Hirsutum hybrid. H into B Barbrans, Arboreum hybrid, hybrid with herbaceum and intra-diploid hybrid. And then there are 2 varieties for which the species was not known. Then hybrid with the CMS3. This was the genetic stock available at CICAR regional session those days, 2008. Then farmers varieties 31, color cotton 1 variety. Now color cotton more than 6. We have the conditions for doing the research, test, conduct of test. 2 season, 2 location. Then for cotton, grouping characters, leaf shape, flower petal color, flower pollen color, ball shape, fiber length. These are the 6 characters we have taken for grouping of the cotton varieties. From that group, we have to select the reference variety, and that reference variety should be grown along with the candidate variety. Then we have a code MGMS VGVS, measurement by a single observation of a group of plants or parts, measurement of a number of individuals plants and parts of plants. Once you see in the field only, you can realize what it is actually. Then visual assessment, measurement by growth habit, symbodial length, plant height, seed weight measurement. Visual assessment, leaf color, pollen color, petal color, visual assessment. Visual assessment by observation of the individual plants or parts of plants, ball size, shape, individual ball shape you have to see. Leaf color, whole plot you can see. VG is the leaf color, VS is the ball shape. So hypocortical pigmentation, we have 2 stages, present, absent. Plant stem pigmentation, present, absent. Stem hairiness, we have absent, sparse. Then palmate, digitate, semi-digitate, and okra leaf. Lobe number 1, 3, 5. Leaf size, we have 3 category. These categories are called states. These states are denoted by the nodes. Leaf cup shape, flat. Leaf nectar is just back. If you see just back side of the leaf, there is a gland called nectar. So present, absent, we have petiole pigmentation, bract type, bract number of serrations, petal spot, position of stigma in the flower, filament color, anther color, ball bearing habit, solitary cluster. Normally, cluster flowering will have determinate plant. In the cluster bearing will be there in the determinate varieties. Solitary fruits will be there in the indeterminate type of varieties. Then ball size, ball color, ball shape, ball surface, prominence of ball tip, then ball opening, ball weight, weight of silk cotton, fiber length, plant growth habit, indeterminate and determinate, plant height, seed first color, seed size, fiber color, fiber strength, fineness, uniformity, fiber maturity. Then ginning percentage, seed density of first. These we have removed 4 characters from this list. Fiber maturity, we are not we have ectocytum cotton, arboreum cotton, herbaceous cotton, and barberines cotton. Then our cotton project, we have some objective. We have kept some objective: establishment and maintenance of database on extant cotton varieties. We have a database for the all available cotton varieties in India. Conduct of dust test of new VCK and farmers varieties. Maintenance breeding of reference collection of varieties. Morphological characterization of cotton varieties. Registration of extant cotton varieties as well as new cotton varieties. These were the 5 objectives we have kept for our project. Characterization of and seed multiplication. So this data from 2014 I am giving, but even prior to that I have data. So this many number of varieties we have taken every year for characterization and multiplication of seed. Then maintenance of reference collection at Coimbatore, G. crucitum, G. barbarans, G. arboreum, G. herbaceum. Continuously every year we have done, but due to lack of space, during 2022-23. This year we will be doing again. Tetraplane diploid cotton varieties under dust trial, dust test at CIC in Coimbatore. So new varieties we have done 50 number last year. VCK and farmers variety EDV, IV. And no varieties were there in the trial, but in the 2014 onwards, we have done varieties with the VCK, farmers variety, EDV and AV. Dust testing at participating centers. We have participating center, CICR Nagpur, US Dharwad, MPK Viraguri, HI Visar and PAU Ludhiana. So this many number of varieties were tested under our supervision. So on the overall progress from 2008 to 2021 is 1,237 varieties we have. Dust testing was conducted, of which around 871 varieties, no, 558 varieties at Coimbatore. Coimbatore, we can see 826 varieties and remaining varieties from the participating center. Altogether, we have done for 1,237 up to 2020-21. So any questions you can ask me, any clarifications? This is the overall and outline. Any in-depth discussion on any specific aspect you can raise the points and we can have a discussion for some time. Thank you for patient listening.
So thank you, sir, for your wonderful lecture. 1 hour of time, you have covered entire dust testing and PPFR Act, the role and everything. We are having one course on dust testing.
Yes,
for first PhD.
They used to come. No?
Sir, you have simplified everything and in your style you have explained.
Actually, in reality, actually, I tell you, once you study this act, you won't get anything in your mind. As a vague, it will go as a vague. Second time, third time, fourth time you study, repeatedly you should read it. Fourth time, if you read the act, then you will feel that there is something. Then only you will feel really, this is happened to me also. Not only me, for many people.
But the whole lecture, no, you simplified everything.
I made simple. I wanted actually.
Made it. They simplified all these things to the understandable way to our students. So
Even,
I have to.
I tell you one thing. Even when we were writing this article, LRA, CV, cotton variety, LRA, CV, LRA 5166. Why CV? Yeah, you see all previous articles. People used to write MCU, CV, MCU5. All our thesis also it is there. And many were not known, including me, that why we are putting CV, cultivar. Why not variety? Cotton variety, MCU5, cotton variety, LRA, cotton variety, Anjali. Once it passes through ACRP, it reaches to the level of variety. Once it comes to the cultivation, it comes to the level of cultivar. So that we were not knowing, and after repeated reading and other discussions with various people only, we come to know that it reaches the, once it is cultivated, it is a cultivar. So that type of confusions were there, many discussions were there. So that was the thing. Any doubt you can ask me. If it is possible for me to answer, I will answer. Otherwise, if I don't know, I will say, I will refer and tell like that. We can discuss, no problem.
Sir, first of all, I wish to congratulate from the bottom of the heart for the very light lecture, sir. It is a very important subject. You made it very light. Sir, it's not a question, sir. I just wanted to know any interesting occurrences with respect to the community rights that was established through this act, sir. Any community was very...
Actually, community right in cotton, especially in cotton crop, the question doesn't arise. Even in cotton, we don't have farmers varieties as of now, right? For paddy, hundreds of farmers varieties are there. Cotton, in the whole of all 142 crops, the number of notified varieties available is paddy, 1,200 and odd. The number of notified varieties in the second crop is cotton. The number of PVP application first in cotton. Number of PVP applications, second in paddy. So this was the situation. So in paddy crop, these community issues came. Actually, TNA also some paddy varieties, farmers varieties were given. Dr. Ranganayaki period, when she was head. And what I could, and that authority fellows, what they said, many varieties were duplicates. Duplicate. Because I will, in my village, I will submit one application from some 50 kilometer, 100 kilometer away. That person also will submit one application. So that black fellows. So like that, all were grown, put together. Dr. Henchinal encouraged many varieties from Orissa. Orissa it came. So, and many were rejected.
What is the response of the private companies, sir?
Very good response. They are interested. They are interested. They saw that they whether first point is whether it is useful or not. They want to have a record of their own varieties first. Second thing is how to use these certificates for promotional activities. In seed packet, you should write protected. First thing, number one. Number two, through extension methods, you tell them. The lacuna in this act also, I will tell you one thing. Once a variety is registered, it should pass the seed standard. Then again, it should go to the VCU cultivation. ACRP system and DUS system cannot be separated first. Many, they talk about that once a variety is registered, we have every liberty. We have, no one can challenge, no one can question like that. It is not so. For a protected variety, when you produce seed and sell in the market, then your seed law comes into force. To satisfy your seed law, you should go for the ACRP trial. Why ACRP? How ACRP generated came in this ICAR system? It is the scheme of DAC, Seed Division. For Seed Act, the nodal officer for implementation of Seed Act is the Joint Secretary Seeds. But the nodal officer for implementation PVP Act is the chairperson. He is equivalent to Secretary Agriculture. But reporting to Joint Secretary Seeds. This was the setup. Seed Act, this Seed Act, implementation of the Seed Act was given to Seed Division of DAC. So they don't have manpower. So they gave the entire, as per our section 5 of the Seed Act, wants that a variety should be tested in over a period in different locations. Multi-location testing is needed. There nothing is mentioned, multi-location testing. Quality seed is essential. Section 5 of the Seed Act will say quality seed. So quality seed, where it will come? From a quality variety. From how we will get a quality variety? It should perform in different places with the same quality. So quality come. That's why that ACRP system came. DAC has no manpower to do. DAC has given the entire project to ICR. ICR kept all the management positions with ICR. All SAs are made partners. So tell me where for any crop, you tell me any university officer is the coordinator. For at least 70, 80 coordination is there, project is there. Invariably, if you say everywhere, that ICR fellows will be the coordinators.
So from my side.
That's why. Even that PVP also, any university staff is the nodal officer for any of the crop. You won't find it. Reason is it was given to NSP. The whole project is given to NSP. Dr. Chowdhury kept as the university CTEC sections are nodal, and in the ICR CTEC section as a nodal, university CTEC department as the co-nodal. So in your department, our seed technology department, many scientists will be working on different crops for dust testing, but none will be the nodal officer for that crop. So this was the reason. So it was kept like that.
Third question, sir. Any interesting court cases regarding this PPA?
Several. Many are there. One interesting is the recent one is the lace potato. One potato variety It's a Pepsi variety, Pepsi company variety, registered with plant variety protection, that potato variety. But Himachal and these Punjab farmers started sowing, started multiplying. And Pepsi went to Delhi High Court. Then it was referred to PVP. PVP cancelled the certificate. You are violating. You are not allowing our farmers to grow your variety.
Farmers' rights, sir.
Farmers' right. Farmers' right. You are violating. You are contending in the Delhi High Court. So Delhi High Court referred to PVP. Dr. Prabhu was chairperson, that previous chairperson. What they said, so we will revoke your certificate because you have violated condition. So this was the latest thing happened.
Thank you, sir.
This is general, you told about this third party
Evaluation.
now, the technology is identified for nowadays in previous period, you said the previous PP was chair period, in the ICR, especially in the ICR institutes, not seed technologists involved.
Where seed technologists are not there. Now also, after my retirement, it will go to a breeder.
Only all the centers breeders are doing this testing, except DNAU. Sugarcane Breeding Institute also breeders are.
Sugarcane Breeding Institute.
DNAU seed technologists are doing. And
Sugarcane Breeding Institute, it was not given to breeder. It was given to actually economic botanist. Initiated with economic botanist. When that botanist retired, given to a breeder because no alternative is not there. Seed people were there. They were Neelamadi and Rajendra Prasad. They were engaged in different aspects.
Not only in SBI, but all other ICR institutes, most of them.
Wherever seed section is there, seed scientists are there. They were given opportunity to lead this program.
Okay.
Once they retire, next best, then breeding it will go. Now also, I made one breeder to associate to me. So once I retired, if no seed technologies come and join, temporarily I will give it to him. Then once a new person comes, then it will be handed over to that fellow. So this was the internal arrangement only. Not like that it should be. Then Dr. Hanchinal made a hue and cry that only it should be conducted by the breeder. Then Joint Secretary Seed called Dr. Chaudhary and asked him to answer a question that why it should not be conducted by a breeder. Chaudhary made a excellent clarification why it was not there. It is there in the file. So from that time onwards, that many centers, scientists were shifted from seed technology to breeding. Like that, some changes were there. So from that time, they stopped it. So this was the thing happened. இல்ல கருங்கண்ணி வந்து டிப்ளாய்டு வெரைட்டிமா ஆர்போரியம் உப்பம் காட்டன்ங்கிறது வந்து ஹெர்பேசியம். தூத்துக்குடி ஏரியாவில் உப்பம், where the coastal area, in Gujarat also coastal area, you will cultivate where soil salinity is more, you can cultivate herbaceous cotton. Karunganni is arboreum cotton. Both are deployed, cultivated especially in the southern part under rainfed condition. They won't follow any spacing. Simply they broadcast the seed. They don't maintain properly. Now they will go for harvest. Nowadays, high density planting one system we suggested. So they follow random sowing instead of random sowing, a line sowing with narrow spacing. They follow that system. Now that system is there. Karunganni usage is after the arrival of BT hybrids, non-BT varieties ரொம்ப கம்மி 5% தான் non-BT. 95% BT cotton variety hybrids are grown. All under the private. நம்ம கிட்ட இருக்கிறது வெரைட்டி பிடி வெரைட்டிஸ் இருக்கு. சிஐசிஆர்லேயும் ஒரு 8 வெரைட்டி இருக்கு. பிடி வெரைட்டிஸ் சீடு டு சீடு. அங்கே வந்து சவுத்தில் இருக்கு. தூத்துக்குடி ஏரியாவில் கொஞ்சம் ஏரியா இருக்கு. ரொம்ப ரொம்ப கமர்ஷியலாக பெரிய அளவில் நீங்கள் பார்க்க முடியாது. உப்பம் காட்டன் அது பேர். நல்ல ஒரு வெளிர் பச்சையாக இருக்கும் பிளான்ட்டை பார்த்தீங்கன்னா. ட்ரவுட் ரெசிஸ்டண்ட்டு, பெஸ்ட் டிசீஸ் எதுவுமே இருக்காது. அதில் காய் பச்சை கலர் காய் குட்டையாக இருக்கும். கிளாப்ரஸ் தான் இது ஹேர்ஸ் இருக்காது. பஞ்சு வந்து ரொம்ப சாஃப்டா இருக்கும், நல்ல ஒயிட்டா இருக்கும், பட் ஸ்ட்ரென்த் இருக்காது பஞ்சில். அது வந்து நான் டெக்ஸ்டைல் அந்த ஆக்டிவிட்டிக்கு அதை யூஸ் பண்ணுறாங்க. கயிறு இந்த மாதிரி ஆக்டிவிட்டிஸ் இருக்கு இல்லையா, பில்லோ மேக்கிங், சர்ஜிக்கல் காட்டன், இந்த மாதிரி இருக்கு. ஆர்போரியும் சரி, ஹெர்சுட்டம் ஹெர்பேஸையும் சரி, இந்த காட்டன் வந்து மெயினாக டெக்னிக்கல் டெக்ஸ்டைல். அதுக்கு பேர் டெக்னிக்கல் டெக்ஸ்டைல். அதர் தான் ஃபேப்ரிக்கு நீங்கள் என்ன பண்ணாலும் அதுக்கு பேர் டெக்னிக்கல் டெக்ஸ்டைல். ஒரு காலத்தில் வந்து இந்த மோட்டாருக்கு ரோப்புன்னு சொல்லுவாங்கல்ல, அதுவே அந்த மாதிரி காட்டன்லேருந்து பண்ணது தான். கயிறு, இப்போ கயிறுலாம் வருது இல்லை, எவ்வளோ மாட்டுக்கு அதெல்லாம் நிறைய கயிறு வருது இல்லை, அது சர்ஜிக்கல் காட்டன், சர்ஜரி யூஸ் பண்ணுறாங்களே காட்டன், அதெல்லாம் வந்து வேக்ஸ் ரிமூவ் பண்ண ப்ளீச்சர் காட்டன். அது வந்து இந்த மாதிரி ஷார்ட் ஃபைபர் அப்சார்பன்சி இருக்கணும், அந்த மாதிரி காட்டனை செலக்ட் பண்ணி வேக்ஸை ரிமூவ் பண்ணிவிட்டு சர்ஜிக்கல் காட்டனாக மாற்றுறாங்க. நிறைய இருக்குது, அது மாதிரி யூசேஜ் நிறைய இருக்குது. பேப்பர் மேக்கிங் இருக்குது, செல்லுலோஸ் தானே காட்டன் இல்லையா, எங்கெங்க செல்லுலோஸ் வருதோ அங்கெல்லாம் ரீப்ளேஸ் பண்ணலாம் இதை வச்சு. இனிஷியல் வெரைட்டி ஐவினா இப்போ எசென்ஷியலி டெரைவ்டு வெரைட்டினா என்ன, அதுக்கு ஏதாவது ஒரு கேரக்டர் வந்து மாறி இருக்கணும். அந்த ஒரு கேரக்டருக்காக தான் நீங்கள் அந்த வெரைட்டியை உருவாக்குறீங்க, ரைட்டா? அப்போ அந்த வெரைட்டி ஏதோ ஒரு வெரைட்டியில் தானே உருவாக்குவீங்க, அந்த வெரைட்டி பேர் ஐவி. இனிஷியலாக இப்போ ஒரு 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 வெரைட்டி, ஏதாவது ஒரு வெரைட்டி பேர் சொல்லுங்கள், உங்களுக்கு தெரிஞ்ச ஒரு வெரைட்டி பேர் சொல்லுங்கள். ஏதாவது ஒரு வெரைட்டி சொல்லுங்கள், உங்களுக்கு தெரிஞ்ச ஒரு வெரைட்டி சொல்லுங்கள். MCV5 இருக்கா? MCV5ல ஏதாவது ஒரு டெஃபெக்ட் இருக்கும்ல, ஏதாவது ஒரு டெஃபெக்ட் உங்களால் கண்டுபிடிக்க முடியும். அது கூட பழகுனீங்கன்னா இதில் இந்த பர்டிகுலர் ஆஸ்பெக்ட் லேக்கிங் அப்படின்னு தெரியும். அப்போ லேக்கிங் போது அந்த பர்டிகுலர் ஆஸ்பெக்டை சாட்டிஸ்ஃபை பண்ணணும், ஃபுல்ஃபில் தட்டு கேரக்டர். அதுக்காக ஒரு மெத்தட் பிரீட் பண்ணுவீங்கல்ல, அந்த ஒரு கேரக்டர் சாட்டிஸ்ஃபை ஆகணும். பட் ரிமைனிங் கேரக்டர் ஆஃப் MCV5 வந்து இருக்கணும், சரிங்களா? அந்த கேரக்டர் எதுவுமே சேஞ்ச் ஆகக்கூடாது, அன்சேஞ்சு. இந்த MCV5 வந்து மற்ற காட்டன் வெரைட்டிகளிலிருந்து டிஃப்ரெண்ட்டாகவும் இருக்கணும், இதுதான் கண்டிஷன். ஸோ நீங்கள் புதுசாக உருவாக்குற ஒரு வெரைட்டிக்கு வேற ஒரு பேர் கொடுக்கணும், MCV5னே சொல்ல முடியாது. MCV5 இருக்கு, நான் ஒன்று சொல்லிட்டா, MCV5 EDV என்னட்டு MCV5 VT. MCV5ம் MCV5 VTயும் ஒரே மாதிரி தான் இருக்கும். அது வெர்டிசிலியன் டாலரண்ட், இது வெர்டிசிலியன் சசெப்டபிள். MCV5 வெர்டிசிலியன் சசெப்டபிள், MCV5 VT வந்து வெர்டிசிலியம் டாலரண்ட். MCV5 பிரெட் வந்து பை காட்டன் டிபார்ட்மெண்ட் TNAயோ, MCV5 VT CICR கோயம்புத்தூர். இதுதான் EDV IVக்கு எக்ஸாம்பிள். MCV5 இஸ் அன் IV, MCV5 VT இஸ் அன் EDV. டிப்பிக்கல் எக்ஸாம்பிள், நமக்கு தெரிஞ்ச எக்ஸாம்பிள் இது. இது மாதிரி நிறைய கண்டிஷன் இருக்கு. ஒரு EDV உருவாக்குறதுக்கு நிறைய கண்டிஷன்ஸ் இருக்கு. லீகலாக நம்மளாம் எப்படி கிளைம் பண்ணலாம்? முதல்ல MCV5 ரிஜிஸ்டர்டு வெரைட்டியாக இருக்கணும், அந்த ரிஜிஸ்டர்டு வெரைட்டிகிட்ட இருந்து அந்த பிரீடர்கிட்ட இருந்து பர்மிஷன் வாங்கியிருக்கணும். தோஸ் டேஸ் நத்திங் வாஸ் தேர். சிம்பிளாக கொஞ்சம் விதை எடுத்தாங்க, போட்டாங்க, யார் வேணாலும். அப்போ பப்ளிக் டொமைனுக்கு வந்தது இல்லை. ஒன்ஸ் இட் இஸ் நோட்டிஃபைடு, இட் கம்ஸ்ன்றது பப்ளிக் டொமைன். ஸோ எனிபடி கேன் டேக் எனி வெரைட்டி, எங்கே வேணாலும் ரிசர்ச் யூஸ் பண்ணலாம். ஃபார்மர்ஸுக்கு ஃபுல் ஃப்ரீடம் இருந்தது, எல்லாம் பண்ணலாம். பட் இந்த மாதிரி லீகாலிட்டி வரும்போது தான் எங்கேருந்து வாங்கினீங்க, எப்படி பர்மிஷன் வாங்கினீங்களா, இந்த மாதிரி கொஸ்டின்ஸ் எல்லாமே வருது. ஆத்தரைஸ்டாக பர்மிஷன் வாங்கலாம், போஸ்டாகவும் பின்னாடியே வாங்கலாம். ஒன்ஸ் அந்த வெரைட்டி ரிஜிஸ்டர் ஆச்சா ஆகலை. MCV5 VT ரிஜிஸ்டர் ஆச்சா, அதுவும் ஆகலை. MCV5 VTயிலேருந்து சுரபின்னு ஒரு வெரைட்டி, அதுலேருந்து கொஞ்சம் பேரண்ட் எடுத்து வேற ஒரு அமெரிக்கன் ஒரு ஸ்பீசிஸ் இருந்து எடுத்து கிராஸ் பண்ணி சுரபின்னு ஒரு வெரைட்டி வந்தது. அந்த வெரைட்டி ரிஜிஸ்டர் பண்ணணும், அது ரிஜிஸ்ட்ரேஷன் பீரியட் முடிஞ்சது. சப்சிக்வெண்ட்டாக வர்ற வெரைட்டிலாம் நாங்கள் ரிஜிஸ்டர் பண்ணிட்டு இருக்கோம். அப் டு டேட் ஆல்மோஸ்ட் ரிஜிஸ்டரிங் வித் 30. எக்ஸ்டெண்ட் வெரைட்டி வந்து, இந்த எக்ஸ்டெண்ட் வெரைட்டி எப்படின்னாக்கா, இந்த PVP ஆக்டுக்கு முன்னாடி என்னென்ன வெரைட்டி எக்ஸிஸ்டிங்கில் இருந்ததோ அதெல்லாம் எக்ஸ்டெண்ட் வெரைட்டி. அந்த எக்ஸ்டெண்ட் வெரைட்டியும் ரிஜிஸ்டர் பண்ணலாம். நோட்டிஃபை ஆன பீரியட்லேருந்து பேலன்ஸ் எவ்வளோ பீரியட் இருக்கோ அந்த பீரியடுக்கு மட்டும் ரெஜிஸ்ட்ரேஷன் கிடைக்கும். அதுக்கு பேர் தான் எக்ஸ்டெண்ட் வெரைட்டி. அந்த இதெல்லாம் எக்ஸ்டெண்ட் வெரைட்டி ஃபார்மர்ஸ்கிட்ட இருந்து தெரிஞ்சது. இந்த செக்ஷன் 5 சீட் ஆக்ட், செக்ஷன் 5 ஆஃப் த சீட் ஆக்ட்னு சொல்லியிருக்கேன். செக்ஷன் 5 எடுத்து படிச்சு பாருங்க, என்ன புரியுதுன்றது. முதல்ல சீட் ஆக்டுடைய செக்ஷன் 5 எவ்வளோ பேர் படிச்சாங்கன்னு தெரியல. செக்ஷன் 5 ஆஃப் த சீட் ஆக்ட் எல்லா இடத்துலையும் சொல்லுவாங்க. ஒரு வெரைட்டி உருவாகிறதுக்கு காரணமே செக்ஷன் 5 தான் பேசிக்காக. அதில் வந்து ஒரே ஒரு வரி சொல்லியிருக்கோம், குவாலிட்டி சீடு மட்டும் தான் சொல்லியிருக்கோம். நீ வெரைட்டி இப்படி பிரீடு பண்ணு, ப்ரீடிங் பண்ணு, மியூட்டாஜன் பண்ணு, டபுள் கிராஸ் பண்ணு, மல்டிபிள் கிராஸ் பண்ணுன்னு எதுவுமே சொல்லியிருக்காது. ஜென்ரலாக சொல்லுவோம் ஒரு வெரைட்டி, ஒரு குவாலிட்டி வெரைட்டின்னு கூட சொல்லலாம், குவாலிட்டி சீடு வரணும் தான் சொல்லலாம் செக்ஷன் 5. அந்த குவாலிட்டி சீடு எங்கேருந்து வரும்? குவாலிட்டி வெரைட்டிலேருந்து வரும். குவாலிட்டி வெரைட்டி எப்படி பண்ணணும்? அதுக்கு நம்மளாம் டிவைஸ் பண்ணுறோம் மெத்தடு. டிவைஸ் பண்ணி கடைசியில் ஒரு வெரைட்டியினுடைய வெரைட்டி ரிஜிஸ்டர் ஆகுதா சீடு ரிஜிஸ்டர் ஆகுதான்னு அடுத்த கேள்வி. இது ஒரு கான்ஃப்ளிக்ட் உருவாக்கலாம். நிறைய பேர் பேசுறது இல்லை. உண்மையிலேயே சீடு ஆக்ட் பிரகாரம் வெரைட்டி ரிலீஸ் பண்ணுறீங்களா, சீடு ரிலீஸ் பண்ணுறீங்களா? சீட் ஆக்ட் எக்ஸ்பர்ட்லாம் சொல்லுங்க. வெரைட்டி தான் பண்றோம் இல்லையா? அந்த வெரைட்டி வந்து எதனால வெரைட்டி பண்றோம்? அது ஒரு குவாலிட்டி சீடை கொடுக்குங்கிற நம்பிக்கையில் அது வெரைட்டி ஆன் அசம்ஷன் பண்ணிக்கிறோம் இல்லையா? ஸோ அந்த வெரைட்டியை வந்து கண்டினியூஸாக அந்த குவாலிட்டி சீடு சப்ளை பண்ணலனாக்க டீநோட்டிஃபை பண்ணுறோம். அந்த எவ்வளோ நாளைக்கு அந்த வெரைட்டி குவாலிட்டி சீடு கொடுக்கும்? 15 வருஷத்துக்கு கொடுக்கும். ஸோ ஒரு வெரைட்டி நோட்டிஃபை ஆச்சுன்னா அந்த வெரைட்டி வேலிடிட்டி பீரியட் வந்து 15 இயர்ஸ். 15 இப்போ ப்ரீவியஸாக இருந்ததுனாக்க இந்த வெரைட்டி ஒன்ஸ் நோட்டிஃபைடு 15 இயர்ஸ் தாண்டிச்சுனாக்க டீநோட்டிஃபை பண்ணுறதுக்கு ஒரு மெத்தட் இருந்தது, டீநோட்டிஃபைடு வெரைட்டிஸ் இருந்தது. இது பெரிய அவஸ்தையாக இருந்தது. எது நோட்டிஃபை ஆகுது, எது டீநோட்டிஃபை ஆகுதுன்னு ஒரு பெரிய கன்ஃபியூஷன் வந்தது. அப்போ என்ன ஆச்சு? இப்போ ஒரு வெரைட்டி நோட்டிஃபை ஆச்சுனால் நோட்டிஃபைடு ஃபார் எவர், நோ கொஸ்டின் ஆஃப் டீநோட்டிஃபிகேஷன், ரைட்? நோட்டிஃபிகேஷன் பீரியடு 15 இயர்ஸ். ஒரு வெரைட்டி இன்னைக்கு ஒரு வெரைட்டி நோட்டிஃபை ஆச்சுனாக்க வெரைட்டி எங்கே நோட்டிஃபை ஆகுது? சப்கமிட்டிக்கு எப்படி ப்ரொபோசல் போகுது? வெரைட்டி ஐடென்டிஃபிகேஷன் கமிட்டி. வெரைட்டி ஐடென்டிஃபிகேஷன் கமிட்டி எங்கேருந்து வருது? ஆல் இண்டியா கோஆர்டினேட்டட் ஒர்க் ஷாப்பில் வெரைட்டி ஐடென்டிஃபிகேஷன் கமிட்டி இருக்குது. அதில் 15 எக்ஸ்பர்ட் இருக்காங்க. அவங்க வந்து வெரைட்டி ஐடென்டிஃபை பண்ணுறாங்க. வெரைட்டல் ஐடென்டிஃபிகேஷன் கமிட்டி. அதுக்கு பிறகு நோட்டிஃபிகேஷன் கமிட்டிக்கு போகுது. சென்ட்ரல் சீட் கமிட்டியினுடைய சப் கமிட்டி ஃபார் வெரைட்டல் கேரக்டரைசேஷன், நோட்டிஃபிகேஷன் ஆஃப் நியூ பிளான்ட் வெரைட்டிஸ்ன்னு முடியும்ல, அந்த கமிட்டி. நோட்டிஃபை ஆச்சுன்னா 15 இயர்ஸ் வேலிடிட்டி. என்ன கவர்மெண்ட்டில் வந்து நீங்கள் 15 இயர்ஸ்க்கு பிறகு நான் அந்த விதையை வாங்கி கொடுக்குறேன்னு கேட்டிங்கன்னா ஒரு சப்சிடி கிடைக்காது, உங்களுக்கு ஃபண்டே சாங்ஷன் ஆகாது. நோட்டிஃபை ஆகி 15 இயர்ஸ் ஆனவருக்கு எந்த ஸ்கீம்லையும் உங்கள் கிராப் ப்ரொடக்ஷன் கைட்லேயே இருக்காது. படிச்சு பாருங்க. சொல்லுமா.
Good morning, sir. Myself Pavitra, second PhD from the Department of Seed Science and Technology. Sir, my question is, sir, generally for agriculture and horticulture crops, we are giving dust testing under 2 seasons under 2 different locations. Sir. For forest tree species, whether they are following the same procedure or?
அதில் ப்ரொசீஜர் வேறு. ஆன்சைட் டஸ்ட் டெஸ்டிங்னு ஒன்று இருக்குது. ஆன்சைட் மதர் ட்ரீ வந்து ஒரு இடத்துல இருக்கும். உங்களுக்கு நீங்கள் 15 வருஷம் ஃப்ளவரிங் வரதுக்கு 10 வருஷம் ஆகும், முடியாது இல்லையா? ஸோ மதர் ட்ரீயை வச்சுக்கிட்டு நீங்கள் எந்தெந்த ட்ரீலாம் என்னென்ன கேரக்டருக்கு ஐடென்டிஃபை பண்ணியிருக்கலாம் இந்த ட்ரீவே சர்டிஃபை பண்ணிடுறது ஆன்சைட்டு. இது வந்து ஒன்று. 2 வந்து இந்த வெஜிடேட்டிவ்லி ப்ராபகேட்டட் இந்த லிம்பு கட்டிங் இதெல்லாம் இருக்கு இல்லையா, குளோனல் ப்ராபகேஷன், அதுக்கு 5 இயர்ஸ் பீரியட் இருக்குது. ஸோ 5 இயர்ஸில் உங்களுக்கு தெரிஞ்சிடும். சிங்கிள் டெஸ்ட்டு 1 சைட்டு அது பண்ணிடலாங்க. அது மாதிரி பண்ணுற ப்ரொசீஜர் இருக்கு.
தேங்க்யூ.
சொல்லுங்க தம்பி. பண்ணலாம். நீங்கள் வந்து இல்லை கண்டிஷன் வந்துங்க, நீங்கள் வந்து உங்கள் வெரைட்டி வந்து டிப்பிக்கலாக இந்த கேரக்டர் இருக்குன்னு நீங்கள் கிளைம் பண்ணணும். கிளைம் பண்ணால் அத்தாரிட்டி வில் சர்ச் ஃபார் யுவர் என்விரான்மெண்ட்டு. அத்தாரிட்டி வந்து உங்களையே கூட கேட்கும், நீங்கள் எங்கேருந்து எடுத்தீங்க, உங்களுடைய ஐடியல் என்விரான்மெண்ட் சொல்லுங்கள், அங்கே டெஸ்ட் பண்ணுறதுன்னு கேட்கும். ஐடியல் என்விரான்மெண்ட் நீங்கள் சொன்னீங்கன்னா ஒன்று அத்தாரிட்டி அதை எடுத்துக்கலாம், அல்ல அத்தாரிட்டி அதுவே சஜஸ்ட் பண்ணணும். அதுவே வந்து நீ இந்த இடத்துல போய் டெஸ்ட் பண்ணணுன்னு மற்ற சென்டருக்கு சொல்லிடுவாங்க. இந்த கண்டிஷனில் டெஸ்ட் பண்ணணும்னு ஒரு ப்ரோட்டோகால் உருவாக்கிறாங்க. அதுக்கெல்லாம் பேர் தான் ஸ்பெஷல் டெஸ்ட்டு. ஸ்பெஷல் டெஸ்ட் என்னென்ன பண்ணலாம் அப்படின்னு ஒரு லிஸ்ட் ஆஃப் டெஸ்ட்டை வச்சு சலைனிட்டி பேருக்கெல்லாம் சாஃப்ட் கேரக்டர் தம்பி, அது பேர் நாட் நோ, விசிபிளி நாட் நோ இல்லையா, சாஃப்ட் கேரக்டர் பண்ணலாம். அதெல்லாம் ப்ரொவிஷன்லாம் இருக்கு. ஆளுங்களா நீங்கள் ஒன்று பண்ணால் ஒரு ரிசல்ட் கொடுங்க, நான் ஒன்று பண்ணால் ஒரு ரிசல்ட் கொடுங்க. பண்ணுறேன்ல. இல்லை தம்பி, ஒரே லேப்ல 2 பேர் 2 விதமான ரிசல்ட் கிடைக்கல, கன்சிஸ்டன்சி இல்லை. இந்த மெத்தடில் பண்ணணும்னு நீங்கள் சொல்ல முடியாது இல்லையா? ஒரு மார்க்கரை வச்சு இந்த மெத்தடில் தான் நீங்கள் பண்ணணும்னு நீங்கள் டிஃபைன் பண்ண முடியாது. அந்த 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 ஃபெசிலிட்டி அந்த லேபில் இருக்காது இல்லையா? எந்த மார்க்கர் யூஸ் பண்ணாலும் நீங்கள் ஒரு மார்க்கர் யூஸ் பண்ணுவீங்க, நான் ஒரு மார்க்கர் யூஸ் பண்ணுவேன் இல்லையா? அது மாதிரி ப்ராப்ளம் இருக்குது. அதனால் இது இன்டர்நேஷ்னலி நாட் அக்செப்டட், நாட் ஓன்லி இந்தியா, இன்டர்நேஷ்னலி நாட் அக்செப்டட். இன்னும் நிறைய கொஸ்டின் வந்துகிட்டு இருக்கு. இந்த பிளட் டெஸ்ட் பண்ணுறீங்கன்னா அதுக்குள்ள ஒரு வேல்யூஸ் இருக்குல்ல, ரேஞ்சு, இதெல்லாம் யார் வைக்கிறான்னு கேட்குறாங்க. அதுக்கு பேர் தௌசண்ட்ஸ் அண்ட் லேக்ஸ் ஆஃப் மெட்டா டேட்டான்னு பேர். அதில் எடுத்து பார்க்கறது, கீப் ஆன் ஆடிங் டேட்டா. நம்ம ஹாஸ்பிட்டலில் ட்ரீட்மெண்ட்டுக்கு போகிறோம்ல, தே ஆர் ஜெனரேட்டிங் டேட்டா வித்தவுட் அவர் நாலேஜ். அது நிறைய பேருக்கு தெரியுது இல்லை. அது நம்ம லேப் டெஸ்ட்லாம் கொடுக்குறோம்ல, சாம்பிள், இந்த டேட்டாலாம் போய்கிட்டே இருக்கும். நம்ம பிளட் சாம்பிள், நம்ம யூரின் சாம்பிள், நம்ம என்ன பண்ணுறோம், எக்ஸ்ரே எடுக்கிறோம், எல்லாமே வந்து அது ஒரு டேட்டா பேஸுக்கு போய்கிட்டே இருக்கும். அது நமக்கு தெரியாது. அந்த அதுக்கு பேர் மெட்டா டேட்டா. அந்த மெட்டா டேட்டாவிலிருந்து ஜென்ரேட் பண்ணுறது தான் பிளட் ப்ரெஷர் வந்து இவ்வளோ இருந்தால் ஐடியலு, சுகர் இவ்வளோ இருந்தால் ஐடியல், இதெல்லாம் அப்படி டிரைவ் பண்ணுறது தான். நம்ம என்னென்ன கேரக்டர் வேணும்னு எப்படி நமக்கு டிசைட் பண்ணோம்? இந்த எசென்சியல் கேரக்டர்லாம் சொல்கிறோம் இல்லையா, இவ்வளோ 30-40 கேரக்டர் சொல்கிறோம்ல, இந்த கேரக்டர்லாம் எங்கேருந்து டிசைட் பண்ணோம்? ஜாம்பிளாஸ்லேருந்து எடுத்தோம். ஜாம்பிளாஸ்ங்க கோர் கலெக்ஷன் ஒன்று பார்த்துருக்கீங்க, கேள்விப்பட்டிருக்கீங்களா? கோர் கலெக்ஷன், ஜாம்பிளாஸில் கோர் கலெக்ஷன். ஜாம்பிளாஸ் நம்ம கோர்ஸில் எங்கேயா வருதா? ஜாம்பிளாஸை பற்றி வருதா? வரல. ஜென்ரலாக பார்த்தீங்கன்னா ஒரு பிரீடர் கூட மூவ் பண்ணிங்கன்னா சொல்லுவார், ஜாம்பிளாஸ் ஏன்டா கோர் கலெக்ஷன் இருக்குது, கோர் கலெக்ஷன் இருக்குன்னு சொல்லுவாங்க. கோர் கலெக்ஷன் என்னன்னா, அந்த கிராப்பில் இருக்க அத்தனை கேரக்டரும் ஏதாவது ஒரு பிளான்ட்ல அந்த கலெக்ஷனில் இருக்கும். சரியா? எந்த கேரக்டருமே விட்டு போயிருக்காது. அது பேர் தான் கோர் கலெக்ஷன். அந்த கோர் கலெக்ஷன்ல இருந்து நம்ம எடுக்கிற டேட்டா தான் இந்த கேரக்டர், இவ்வளோ கேரக்டரும். அதுதான் கண்டிஷன்.
So thank you, sir, for spending your valuable time with us. You patiently listened our queries and happily answered for all the queries in the simplified manner. So I think this deliberation that will be very useful for our students, sir. Thank you very much, sir. You are spending this valuable time with us.
Thank you all for the opportunity.
So,
thank you very much.
now I request all the students, please stand up and give applause to our sir.
Thank you.